So my name is Joshua Goff. My project involves carbon capture and utilization the algal way, effectively using algae for carbon utilization. So before going any further, I have to acknowledge Orlando Utilities Commission, OUC, none of this project would have been possible without them. Essentially what happened was they took their flue gas that they're producing, it's a coal-fired power plant that they have running at OUC, and they produce a flue gas from burning the coal. The flue gas they produce is then being used, it's being scrubbed by sulfur scrubbers and cooled, and that is then injected into algal race clay ponds, which we have on site. There's all I'm going to explain soon. But those ponds are then, the algae we then harvest it ourselves, and that algae is the fundamental of this experiment. But as far as carbon capture goes, we're taking the flue gas that they produced and we're feeding into the algae as a source for CO2, and that algae that's been grown off that is what we're using for this experiment. And before we go any further, I have to say this line, we've been saying it all summer, but the idea that we want to take with this experiment is to take that fossil fuels is fossil thinking. Just off that, fossil fuels are the way of the past. If you want to move to the future with a more renewable source of energy, we have to leave that past behind. And frankly, there's no time like the present to make that positive step forward. This is the shocking realization we live in. This is electricity. It's the breakdown of how electricity is generated in Florida. As you can see on the graph, 3/4 of electricity in Florida is actually generated from fossil fuel burning to some extent. Be it coal, natural gas, or petroleum, 3/4, more than 3/4 actually. And considering that Florida is actually the second largest total net electricity generation in the country, we're a model of how things need to change. If we make a change, the rest will follow. So why is it concerning to have so much fossil fuels being burnt? Well, it's releasing CO2 in the atmosphere. The CO2 released from burning fossil fuels is having a negative impact on our Earth's natural carbon cycle. Ordinarily, the way CO2 should work is it should constantly be going in an endless circle between the atmosphere, the ocean sources, and the crust. However, by mining coal or by extracting oil or natural gas from the actual Earth's surface or below the Earth's surface, you're offsetting the natural cycle and causing too much CO2 to be released in the atmosphere, resulting in too many greenhouse gases floating around. The Earth will start warming. We've all heard of global warming. It's real. So moving on to flue gas again. Like I said, coal-fired power plants, burning coal, releasing flue gas into the air. The flue gas composition is what we see here. As you can see by my lovely graph, carbon dioxide is by far the largest greenhouse emittance. This isn't greenhouse gases. Flue gas is generally between 11 and 15% CO2 in overall composition. So after you burn the coal, if you can take that 11 to 15% CO2 and make a positive use from it, then we're making a huge difference overall. By repurposing that flue gas to feed the algae ponds, we're providing a food source, well, a photosynthesis source for the algae to undergo if necessary processes, and we're also reducing total CO2 emissions in the atmosphere. So it's a double win. It's a double win, really. Now on to the real project. My goal is, of course, to take that flue gas in the atmosphere and find a way to change it into a renewable fuel. And as you can see, here's the pictures of ponds at OUC. There's 4 total ponds at OUC. I need to say that now before I go any further. So the steps involved are, of course, to take the flue gas, feed into the ponds. The ponds will run off the flue gas and the algae will be produced. Then we harvest the flue gas, we harvest the algae later, and the algae we harvest from those ponds is, again, the basis of the experiment. It's how we're going to produce our renewable fuels. But let's get into how that's going to work. So the methodology I have can be broken down into 3 major steps with multiple steps beneath it. The first step I need to do is to characterize the actual algae within the 4 ponds. To do that, I took total solids readings, volatile solids readings, and chemical oxygen demand. The main purpose of all those things was to more or less see, well, total solids and volatile solids to determine how much organic composition was within the actual algae themselves between the 4 ponds. As far as chemical oxygen demand, that tells you how much oxygen is actually needed for methane to be produced. Blaise mentioned earlier as far as theoretical methane production. This is the number we use to do that, so it's a necessary test to determine our theoretics before we do the actual experimental process. I then used light microscopy to actually look at the physical algae and see what I was seeing, see it in a smaller scale. I looked at it at 25x, 40x, and 100x magnifications. The goal was to just see which one of them would be the best picture so I could identify the algae and determine if there was any potentially cellular characteristic of the algae that could impact the actual production of methane overall, or renewable energy. And then the bulk of the project was, of course, to determine the methane index potential, or how much methane was produced from actually processing the algae through an anaerobic digester, Although in our case we did triplicate batch assays, Blaze actually showed you guys a demonstration of how we do the methane production in the lab. So I'll spare you the details, but effectively it's a airtight container that we just take they show you the needle process, it flows through this KOH barrier solution, and we just measure displacement, and that's how we know how much methane we're producing. Blaze also said this, but again, it's very necessary to know this, that it's got the 35 degrees Celsius. Mesophilic conditions are important for methanogens, which are the microbes within the actual inoculum of the serum bottle, which is what we're using for the anaerobic digestion to function at maximum, at ideal conditions. So we keep them at 35 degrees Celsius incubation to maintain that ideal condition. Also, one really important thing is there was no pre-treatment involved, which means we didn't crush the algae in any way, we didn't stir it, we didn't blend it, the walls were no way disrupted. The algae that went into the methane index potential readings was completely untouched. It was from the ponds. We weighed out the necessary loading rate, which means we weighed the amount that would equate to 2 grams COD per liter. Blaze touched on that as well. We weighed out, no touching, all 4 ponds, no treatment. So the values you see here are completely natural, no processing.
In other words, like just for obvious, we don't know what sort of pre-treatment we may need for the feedstock in the future, but the moment we want to establish a baseline,
it's all natural. Literally, it's natural. But here's the results of characterizing the algae. From the start, this is our very first thing we went to do, is we wanted to figure out the organic content. Orange and blue because UF. As you can see from the blue readings, the volatile solids of pond 1, which was microalgae, which I'll get more into soon, demonstrated that they were actually considerably lower than the volatile solids that we saw in all 3 filamentous ponds, pond 2 through 4. So from the start there, we knew that there was a far higher volatile solids or organic composition in the filamentous algae compared to the microalgae. And that's organic solids are necessary for methane production. They're what fuels methane production. So just from here, we had a pretty good idea of, well, I guess it's going to go pretty well. The next line in orange, our other gator color, is the chemical oxygen demand, which, as you can see again for pond 1, is also considerably lower than what we see from pond 2 through 4, which, establishing theoretical methane potential, you have to take the numbers we get from our CODs and use that through the equation to determine how much methane there can be produced. And as you can see, just from the start, microalgae is already falling behind. And the next stage of the experiment was, of course, to look at it through a microscope and see some nice, pretty pictures. To the left here, we see microalgae. There's huge speciation diversity. That's not what I'm going to focus on this thing. This one here is particularly Pegistrum. It's the most prominent microalgae we saw actually in pond 1 was probably this species. But what I want to take away from this is when you look at the cellular structure of microalgae and the cellular structure of filamentous algae, there's some notable differences. Microalgae is clumps. Not so much, well, in this particular case, yeah, it's clumps. But as opposed to it, filamentous algae has far more readily available. The walls could be easily broken. I've seen myself in microscopic observation that the walls of filamentous algae break far easier. The material just escapes. So when the microbes in the digestion process are trying to break through those walls to get to the good stuff, the proteins and lipids that are locked inside, it's far easier to break through the walls of filamentous algae as it would be for microalgae. So that may have a considerable explanation for why we have the results that I'm going to show you now. So here's the big slide that I'm kind of trying to build to. This is the cumulative methane yields of the OUC ponds 1 through 4 across the 28-day incubation period that we used. As you can see through these graphs, these 3 are the filamentous ponds, and they produce far higher methane yields than what we see from the microalgae pond.
And pond 1, remember, is the fishy one.
Pond 1 is the fishy one. Pond 1, tying back to the fish experiment, is the one that had the worms in
the
it.
wormy one.
But in this case with MIPs, we removed the worms prior to actually doing it, so they are not a contributing factor to this experiment.
Well, thank you for my correction.
As you can see, similar to Blaze, that we saw roughly a 2-day lag period. Anaerobic digestion takes time for microbes to acclimate to the environment to get their processes all up and running. But after it took off, it really took off. The 3 filamentous ponds demonstrated very similar yields, especially pond 3 and 4, which were the 2 ponds that were fed on flue gas, as opposed to the other 2 which are fed on pure CO2. But based off the results we see from that, pond 2 through 4, far higher methane yields. The range is somewhere between like 390 and 460, while pond 1 total cumulative yield. It's still climbing, but it's not getting. By the time these are done, this one will maybe be getting somewhere close to it. But overall, the yields are, as you see, very different from microalgae versus film matches. And my conclusion from all of this is that algae grown in flue gas definitely has potential to be used as a renewable energy source. And specifically, filamentous algae, as was shown here, has a significantly higher methane potential than microalgae. So based off of that, growing filamentous algae on flue gas, double 2 wins right there. You can lower CO2 emissions overall, which is important, especially in the case of coal power plants. I didn't touch on this yet, but due to the Clean Power Act of 2015, there's currently a ban on new coal power plants starting up, and coal power plants that are currently going have to decrease their emissions by somewhere between 31 and 32%. So if they could find a way to decrease CO2 emissions through using algae to produce both a positive renewable energy source, while still taking CO2 that would ordinarily be in the atmosphere and use it as a growth for an organism, algae, reducing emissions, and it's providing a new renewable source.